second lecture of module 1, we will talk about amino acids and their properties, and further to the types of amino acids that we learnt in the last lecture, we will see what further characteristics are there of these unique building blocks of proteins. To understand these, we have listed a set of properties: the isoelectric point, an amino acid titration, which is going to help in an understanding of how we look at amino acids, what their structural properties are, the hydrophobicity of the properties of the amino acids, and the spectroscopic properties, which we will do in the next lecture. Now, to look at amino acids, we understood what a chirality meant, the polar and the non-polar parts, and most important, the hydrophobic characteristics of these amino acids. In the general properties of amino acids, when we look at what we call the zwitterionic form, where we know that this is a neutral amino acid. Why? Because it has an NH3 plus and a COO minus. Now, this zwitterionic form is the most common form that is going to be present at the physiological pH of 7.4. We look at the size and the shape, because these are going to be important in how it interacts or how it appears in a protein. We are going to look at the charge, the polarity, the hydrophobicity, the aromaticity, the conformation that is usually determined by the side chain, and the propensity to adopt a particular conformation. Now, as we go on in these different properties and see how they affect proteins in general, not only in structural characteristics, but definitely in functional characteristics as well. And of course, their relative position in the proteins is going to depend upon the type of protein that we also look at. The 20 standard amino acids that we looked at in our class last time was that we had the non-polar or the hydrophobic type, and we had the polar or the hydrophilic type, where we have an aliphatic aromatic set, or we have an acidic, neutral and basic set. If we go into further details and try to understand what these structures are all about, then we see that in the aspartic acid and the glutamic acid, that are the acidic amino acids, we have here the drawing that we see here, or the picture that we see here. We see that there are larger number of red atoms, meaning there are larger number of oxygen atoms, simply because these are the acidic amino acid, which means that apart from the NH3 plus group that every amino acid has, apart from the CO minus group that every amino acid has, this has, in addition to this, a side chain that is also an acidic amino acid. Now, when we look at this particular set, we have to try and understand that this plays an important part. We look at the basic amino acid residues, and we see that when we look at these residues, these are going to help us in trying to understand what we mean by the side chain residues. What are these side chain residues? These side chain residues are going to be those that are going to give us an additional nitrogen in them. So, whether it's histidine that has an imidazole group, whether it's lysine that has the epsilon NH3 plus group, or the arginine that has a guanine group. So, we see the extra nitrogen atoms here. Now, how does this help us in understanding? We will see as we go along, not only in the interactions, but also in very important aspects of proteins. So, when we look at the amino acids, we talk about the alpha carbon, but there are many other chains or many other atoms, moieties that are attached to this central alpha carbon, and we have to look at them in terms of a nomenclature, in trying to understand in a universal fashion how we depict these amino acids in general. So, if we look at the lysine that has been drawn here, we can see that there is a C alpha carbon atom, then there is the C beta, the C gamma, the C delta, the C epsilon, and the NH3 plus, which is the side chain of the amino acid. So, when we look at the nomenclature, or we look at the way they actually form, this is what is important in an understanding of the amino acid characteristics or the amino acid depiction and the way we name them. When we look at now the acidic and basic properties of amino acids, we have the NH2 group, we have the COOH group, and we know in the zwitterionic form this is going to remain as NH3 plus and CO minus. What does this mean? This means that this proton, these protons that are present can be lost. The loss of a proton means there is a particular reaction that can occur that is going to tell us or take us from HA to H plus and A minus, where we have a conjugate base and we have the loss of the proton. Now, this loss of the proton, using the law of mass action in an equilibrium setting, we can find out what the value of the Ka value is, and from the Ka value we get what is called a pKa value, and from that, with the concentration knowledge of the A minus, that is the conjugate base, and the HA, we can find out at a definite pH what is this ratio going to be. Now, how is this going to help us? What can we learn from this understanding? So, when we look at an idea of this particular setting where we are losing a proton, we are trying to look at how the proton is affected, how the amino acid is losing this proton, we will look at a definition, the isoelectric point. This is the pH at which the molecule carries no net electric charge, and we will see how this is important later on when we study more about proteins and their overall distribution of charges, their overall characteristics and their overall interactions. So, if we use this Henderson-Hasselbalch equation, we know that each amino acid has an amino group, has a carboxyl group, and what we are trying to do is we are going to try to find out this isoelectric point that is actually the average of the pK1 value, that is the pKa of the carboxylic group, where the COOH, this particular proton, is lost, and the pK of the amino group, where we have the NH3 plus, where we know this proton is lost. So, because we know that every amino acid, we know we have the C alpha, we have the H, we have the NH3 plus, and we have the COOH. So, this would be the characteristic of an amino acid at high acidic condition, which would mean that all of the possible places or all the possible atoms are protonated, and this R group will then add additional acidity or basicity to the amino acid, which is what is important. So, if we try and understand this further now, and then which will take us to the next set of an understanding, then we will have to see what we mean by these basic characteristics. So, let us look at the next set and try and understand what this amino acid characteristic is. So, we have the dissociation of the first proton from the alpha carboxyl group. This is going to be followed by the dissociation of the second proton from the amino group, and this is common for all amino acids. So, if we look at the simplest amino acid, glycine, it has a plus charge. Where is this plus charge from? It is from the NH3 plus, because the COOH, that is at the lower pH, does not have a charge to it yet. Now, as we have the loss of the first proton, the COOH is going to lose its proton and become CO minus in forming the zwitterionic form. So, we have a K1 value associated with the loss of this first proton from the carboxyl group, which is associated with a pK1 and a pH. Similarly, if we lose the second proton from the amino group this time, which is NH3 plus going to NH2, we will have a similar expression relating to the K2, and we will get a pH value for this also, getting a pK2 plus a log glycine by the glycine minus by the glycine 0. Now, when we look at this, so this is the structure of glycine. If we know that it has 2 hydrogen atoms to it, the simplest amino acid. We have pKa1, we have pKa2. Now, when we look at these values here, so the first loss is going to be this proton, the second loss is going to be this proton. So, it has gone to NH2 and this CO has gone to CO minus. So, what does this tell us? This tells us that the Pi is going to be the average of the pKa below neutral to the pKa above neutral. And what is neutral? 0. So, we look at pK1 plus pK2 divided by 2, that is going to give us the Pi of glycine. Fine. So, now we are looking for a pKa value below neutral, a pKa value above neutral, and that is going to tell us the Pi of the protein. So, now if we look at the titration curve of glycine, this is what it looks like. We have the first break where we have the H loss to CO minus, forming what is called the neutral or the zwitterionic form here. This then goes on to lose its next proton. So, NH3 plus going to NH2, giving us the final form of this species in the titration, where we are losing the H plus ions as we go along by adding, say, sodium hydroxide. So, when we look at the titration of amino acids now with ionizable side chains, this is going to make it a bit different. Why? Because the acidic amino acids are going to have, in addition to the general NH3 plus and the COOH that is associated with every amino acid, they are going to have an additional side chain. So, this side chain of the aspartic acid that we are looking at also has a proton. So, that is also going to be lost when we do the titration. So, now the first proton loss, depending on the acidity or how easily the proton is lost, we are going to first lose this proton. So, at the first instance pK1, we lose this proton. At the second instance, which is the pK of the R, meaning the pK of the side chain, we are going to lose this proton. Now, we see we have lost this proton, and at the end, at the pK2 value, this corresponds to the NH3 plus, which is going to be NH2. So, what does this mean? This means that I have to go from below neutral, I have to go from, say, a charge of +1 to 0 to -1, and the pKa values that are associated with these changes, the average of those is going to give me the Pi of the particular amino acid. So, this is the way we look at it. We have aspartic acid, we have a pK1 value. Now that we understand this, we have a loss of this proton first, then a loss of this proton, then a loss of this proton. So, we know the overall charge. The overall charge here is +1, the charge here is 0, the charge here is -1, the charge here is -2. And what did we learn? We need to get the average that is going to take us from +1 to 0 to -1. So, we need this pK value, we need this pKr value to tell us what the Pi of aspartic acid is. So, this is a simplistic way of looking at the titrations. So, we are adding equivalence of OH minus, we are looking at a titration where we have a pK value here, a pK value here and a pK value here, and we realize, we understand what this is for. So, now we have a Pi that is going to be the average of the pK1 and the pKr. Similarly, if we do something for lysine, we look at the point here. Here we have a charge of +2. Here we are losing the proton here. Now, we have a charge of +1. Now, when we look at the second set, when we are going for the pKr, we have now lost this proton, we have a charge of 0. Now, when we go to the next case, we have a charge of -1. So, what am I interested in? +1 to 0 to -1. So, I have to look at the average of the pKr and the pK2 in case of this basic amino acid lysine. Similarly, we can look at other amino acids as well. The interesting one is histidine. The histidine titration is interesting simply because if we look at the pKa value, it is 6. Now, why is it interesting? Because the pKa value is going to change, or it can change depending upon how easily the proton is going to be lost from the side chain. In this case, it is our imidazole group, which is this. So, when we are looking at the imidazole group that is connected to, or which is the amino acid histidine, the most interesting one is the amino acid histidine. Now, why is this important? This is important because the pKa value of this particular side chain of the imidazole group has a pKa value of 6. Now, the pKr of the side chain depends upon its environment, and what we will see later on when we go into the depths of understanding of proteins, we will see that histidine occupies the active sites of many enzymes. So, it is important to see the role of this histidine in terms of a titration. So, if we consider the imidazole of the histidine at the surface of an enzyme, we will see that the pKa value is 6. Now, what does this pKa value correspond to? It corresponds to the loss of this proton of the imidazole group. Now, we just if we look at a rough representation of the imidazole, looking at it on the surface of an enzyme. Now, what happens if this amino acid is at the active site? So, here we have a rough sketch of a protein, and here is the imidazole group in the active site of a protein. So, it can lose its proton, but will the loss depend upon what is surrounding it? Yes, it will. Suppose we say that there is an abundance of positive amino acids close to this histidine. It will not want to hold on to the proton because that renders it a positive charge. So, it would rather release the proton at an earlier point in the pH, and then what would happen is the pKa value would be less than 6. In the same way, if we say that this is surrounded by negative amino acid residues, in abundance we have acidic amino acid residues, it would rather hold on to the proton and prevent its loss as much as possible, rendering a pKr value that is greater than 6. So, when we have a greater than 6 value, we can have a less than 6 value, and the interesting part here is if you notice the range of this covers our physiological pH of 7.4, and we will see later on during the course. It is interesting to note that the variation in the histidine can go from 5 to even close to 8. So, that is an important feature which we will understand later as we go along. So, if we look at the acid base chemistry of the amino acids, we saw how glycine behaves, we saw how an acidic amino acid behaves, and we saw how a basic amino acid behaves. So, we know now how to calculate the pi of an amino acid, and later on when we study the peptide bond, we will understand how we can actually look at a peptide bond or calculate the pi for a series of amino acids. The next very important property is the hydrophobicity of amino acids. Now, this is extremely important in understanding where an amino acid is going to be comfortable in a folded protein, which we will understand as we go along. But the property in trying to understand what is present in the side chain of these amino acids, we learned that the hydrophobic amino acids preferably have only carbon and hydrogen to them, and a measure of hydrophobicity is given by a hydropathy index. There is a common index known as the Kite-Doolittle hydropathy index, which gives us, as you can see, positive values for hydrophobic, highly hydrophobic amino acids, isoleucine, valine, leucine, which are the hydrophobic amino acids, meaning their side chain contains only carbon and hydrogen with no heteroatom, that is no nitrogen, no oxygen and no sulfur. As we go on to the other types, we will see the values gradually decreasing, saying that their hydrophobicity decreases, and as is expected with the acidic and the basic amino acids, we are going to have high negative values. Now, how does this help us? This helps us in a way that if we have amino acids one after the other in a particular series, which we will see, we can actually identify hydrophobic regions in proteins, and this is commonly used to identify transmembrane regions. We will look at membrane proteins as well, following transmembrane regions. So, we have what is called a method called a sliding window approach, which we will just address in a simple manner, looking at a specific window size. We will explain what this means in a moment. Calculating what is called an average hydropathic index value of the amino acid residues, and we plot this. Now, from the positive and the negative region of the plot, we can determine the surface and the core regions for proteins. So, we know when we saw the table that a positive regions means we have a hydrophobic region, and a negative region would mean we have a hydrophilic region. So, let us look at what this looks like. We have the sliding window approach, 7 or 9 residues, and we look at the hydropathy index, we look at a window size of 7. Now, a window size of 7 means we have 1, 2, 3, 4, 5, 6, 7. We calculate the average of the hydropathy index values from the table that we showed, and we assign it to the middle residue here. Then we slide the window, as I will show you, and then we will see what this looks like. So, say we have a sequence like what is given here. What are these? Isoleucine, leucine, alanine, valine, serine, aspartic acid, threonine, methionine, glycine. So, there is a mixture of acidic, neutral, but polar and hydrophobic residues. Let us see what the characteristic of this string looks like. So, here we have the string, and the particular hydropathy index values have just been assigned to them from the table. And what we do is we look at the first 7 in this case, and we add up the values, calculate the average because we know our window size is 7, and we say that the value is 1.33, a positive value. Then we look at the sequence again. What we do is now we look at our specific amino acid, but now we have to slide over 1 because it is a sliding window approach. So, we still have 7 residues in our window. What do? We again calculate the number, the total number, then divide by 7 to get the average, and we still see that the value is 0.95. And then we go on to do this for the next set, and we keep on doing this till we cover the whole protein, right? Now, when we do this, what we are actually trying to look at is whether we have a positive region. Now, what is this positive region going to tell us? The positive region is going to tell us that we have a hydrophobic region in this case, because if we look at, see, there is here an acidic amino acid, but the overall setup or the overall region that we are looking at actually is hydrophobic in nature, despite the fact that we have a polar amino acid and an acidic amino acid. Now, the window size of 7 or 9 or 11 is sometimes chosen because we can assign the value to the middle residue, the residue in the middle. We do not want too small a window size because that is going to create noise in our plot, and we do not want it to be too large because then we might overcompensate for the values and not be able to get an idea of what this particularly looks like. So, here we have the amino acid composition for 20 amino acids in transmembrane and globular proteins. So, what does this look like? This looks like two proteins, ATPase, which is a membrane protein, and lysozyme, which is regular globular protein. So, if we try to look at the different types of amino acids that we have here and where they are located, we see that we have alanine. Let us look at the larger ones here. We have arginine here, we have alanine here, and we have asparagine here. But if we look at the blue ones where we have valine, we have serine, we have leucine, we have isoleucine, these are the ones that are hydrophobic in nature. So, the hydrophobic nature tells us that this is preferably a transmembrane protein. So, what we learned here was the amino acids, their chirality, the polar non-polar aspects of them, and the hydrophobicity, and an understanding of how we can calculate the pi, the isoelectric point of a protein. What is this isoelectric point? It will tell us, taking us from a charge of +1 to 0 to -1, telling us that this is important. And when we looked at histidine, we found out the importance of histidine in terms of its environment as to how easily it is going to lose the proton that is associated with the imidazole group of the side chain, that is histidine. These are the reference books. Thank you.